cool. So our next presenter is Brendan. He's returning to finish up his talk on the REBUS model for brain action of psychedelics. So Brendan has a PhD in synthetic biology, a field at the interface of bio and engineering. He has a long-standing interest in neuroscience, which he studied in his undergraduate, with a focus on gene-environment interactions encoded epigenetically. He presented on the promise of psychedelics as a therapeutic tool for a research project in his undergrad, but he never imagined that so much progress could be made since then. And so with that, Brendan, please take over.
All right. Thanks so much. Really appreciated being back. Yeah, it's a huge paper. It's not my paper, for those of you who haven't tuned in the first time. It's by Friston and Carhart Harris. It's a tour de force, a massive synthesis of all sorts of different things. Yeah, sorry again that I don't have video. It's because my laptop has decided that its camera doesn't exist. And so I'm just going to be this disembodied voice for the presentation. All right, can you guys see my screen? Leo, can you see my screen? All right, awesome. Okay, so this is the introduction to the paper. This is the table of contents. And last time in my full talk, we covered this much, just the introduction. But there's a lot of ground to cover, right? And if you didn't tune in for that talk, I highly recommend doing that because what's coming next will make less sense if you don't, but obviously you don't have time to do that now. So with that in the bag, I'm going to do a brief summary of that before I begin, and then we're going to move on to the basic neuropharmacology, which we talked a little bit about. Asher just did that. Then how this turns into rhythms networks, how plasticity plays a role, the behavioral evidence for this really interesting addition of insight and how that plays a role, and then we're just going to wrap it up. There's not enough time to cover everything here. All the other sections I don't highlight, but these I think are the most important, at least for this talk. Okay, so let's dive into the background in neuropharmacology. So if you remember, last time I talked about hierarchical predictive coding. This is the system inside the brain in which the brain organizes its processing in different layers. And we talked about how Bayes' theorem, the brain operates using Bayesian principles, and together this hierarchical predictive coding and Bayesian principles make up what's called the Bayesian brain. And the free energy principle, which is a very general principle about how homeostatic systems operate, encapsulates the Bayesian brain and also information theory. And information theory is a very fancy area of math. It has a lot of applications. It's also shared by entropic theory, which then has measurable predictions. So we can connect this Bayesian brain hierarchical predictive coding to measurable predictions, and then we can look at the effects of psychedelics on this. This is the REBUS model. We take this abstract or fine scale mechanism and we turn it into large scale mechanisms that we can measure. Very, very powerful model. But of course, it's what I presented before was just a model. It wasn't actually any of the evidence, which is what we're going to be covering today. And so for a recap, this is figure one, right? I spent most of my presentation just talking about how we get to figure one. Here's the hierarchical predictive coding. We have bottom-up error predictions coming from the sensory, and we have top-down predictions that are ours. And having strong priors means that you can't shift your opinion very much with new information. Any new information that comes in is very much biased by your strong prior you have before. Whereas when under psychedelics, you have weak priors. This means sensory input has much more importance, and we can weaken our priors, we can actually change our minds. We can represent this prior, these very strong priors within a free energy landscape. This is kind of just the whole connection of the brain, the energy that's going on in there. And when you have very strong priors, you're stuck in what are called these minima, right? So at the bottom of this free energy landscape here, there's a minima, and this is predicted by the free energy principle. And you might not be in the lowest minima. The lowest minima would be better for you, but because you're strong priors, you're stuck in a minima. Whereas in psychedelics, this is flattened. This flattened energy landscape allows you to move around and explore different minima. And just as another graphic, as a reminder, in the normal state, you're in a state where your brain's connected in a certain way. Certain regions are connected to certain other regions, and it's more orderly. But in a psychedelic state, you're in, everything's connected to everything else. Not totally, but it's much, much more connectivity, much less order, much higher entropy. I'm going to walk through the final example I gave last time, which is how the hierarchical predictive coding in the Bayesian brain actually ends up changing an experience. So normally we start off with a very, very strong prior. And just as a reminder, each of these levels, these bars here, are different layers of the cortical hierarchy. And information from the top layers percolate down each layer of the hierarchy, right? So at the very highest state, we start with very abstract principles, like this is what I believe. You have a very strong prior here, and any incoming information has a very small change because of such a strong prior. And what under psychedelics, what happens, that I explained before, is that these priors get weakened. And when these priors get weakened, the suppressive effect works its way all the way down And so each layer of the hierarchy becomes less and less biased. And what this does is it allows your sensory information to percolate in more strongly. So when we have initially weak sensory information of you did a good job, you worked really hard, now we can actually much more appreciate this because the feedback we're getting is allowed to happen because the priors are weakened, right? And so we go from this very deep energy landscape, experience a very large change, and then we go to a flattened energy landscape. This is what I described before. What we're going to see is when we instantiate this in a biological system, it's slightly different and it's even more interesting than that. So how do we take this mathematical model and actually turn it into the brain? Is this just a theory, or is there actually strong biological evidence for this? And it turns out that there is. And to get into this discussion, we first have to talk about a little bit of neuron anatomy, okay? This is a classic neuron image. This is actually a pyramidal cell. We're going to talk about more of this. The main thing to note that you'll notice about this, if you've never seen a picture of a neuron before, is it's got all these long spindly things. This is the cell body, and most of this image is just these long spindly branches. These are called neurites, and the majority of these neurites are actually dendrites. Dendrites feed information into the cell body of the neuron. All these connections are feeding information in, and only a single axon leaves the neuron. Because if we zoom into one of these dendrites, we see this covered with all these tiny little spines. These are called dendritic spines. Each of these spines contains at least one synapse. And what's happening is that neighboring neurons, or neurons that can be very far away, they're sending axons, send outgoing information from that neuron. It crosses the synapse and then becomes incoming information for this neuron here. So this synapse, this gray here, is this side that you're seeing here that turns into the green arrow that goes to this neuron. And there's neurotransmitters that cross this synapse. And psychedelics act like neurotransmitters. They bind to different serotonin receptors, and specifically the 5-HT2A receptor, which Astrid just talked about. And this is another crystal structure of the 5-HT2B receptor, similar enough to the 5-HT2A for our purposes. And you can see LSD bound in there in a cutaway image. This is how it all works, right? This is our basic neuron anatomy of how the neuron fires. And the 5-HT2A receptor is important. Asher talked about that it is the primary receptor for psychedelic action. And indeed, if we look at the occupancy of this receptor on the bottom here, so 20% occupancy, 40, 60, 80%, we see that very non-linearly, it grows very quickly, the intensity rating of someone's trip. So this is good evidence, behavioral evidence, that the higher we occupy this receptor, the more intense the trip rating. These receptors are the primary receptor for psychedelic action. And this is further supported by the fact that these 5-HT2A receptors are located in the highest association cortices of the brain. So we know that the brain is hierarchically organized. We know that certain parts of the brain have to do with more abstract processing than others. And what we see here is that the 5-HT2A receptor is localized to those exact parts of the brain that have the more higher executive components of functioning, the conceptual, the abstract thoughts, those kind of world building parts of the brain. And located in those world-building parts of the brain are what are called, there's lots of different types of neurons, but a main type of neuron that I just showed you before is called the pyramidal cell neuron. So what you're looking at here is what's called the Brainbow. Brainbow, you fluorescently genetically label neurons, and they all appear all different colors with different recombination of fluorophores. And you'll see that they do look kind of like pyramids. These little tiny triangles here, these are the pyramidal cell neurons. And you might notice, it's not as clear in this image, but there's different layers here. You can see there's kind of like one layer here, it's kind of like a gap, and there's kind of like another layer here. These are called lamina in the brain. And so we can translate this hierarchical predictive coding map into a laminar layer. I'm going to go into more detail here, okay? So these are the different laminar layers. This is a 6-layer lamina. And what they talk about in the REBUS models, they talk about the layer 5 pyramidal neurons. These neurons located in layer 5. These actually are the pyramidal neurons. They've been in this model the entire time. I've just never talked about them because there's always other labels here and we weren't getting into it. But these neurons are the pyramidal cell neurons located in these areas of the brain. So we're already populating this more abstract model with more concrete biological data. And we can go even further. These are different parts of the brain. This is an agranular cortex, dysgranular granule. We're not going to worry about exactly what these are right now. Suffice to say that you can see that they differ slightly in their laminar structure. There's different numbers and they're kind of compressed. And more importantly, we can see that there's these prediction neurons, right, that send down the predictions, right, these strong priors. And then there's prediction error neurons. These prediction error neurons send the prediction errors. And you can see that they're all connected to each other. It's complex, but we're gonna clarify this in a little bit. And so we can now populate this model even more specifically. On this side, we have the prediction error neurons in red. And on this side, we have the prediction neurons in green. So now these aren't just pyramidal neurons. We can now distinguish between prediction error pyramidal neurons and prediction neurons. And if we take this stratified kind of like cut crossway section of the brain to different laminar layers, and we translate this into actually the brain anatomy, we can see that these different cortices lie in different cortical areas in the brain. So here we see the green, agranular, occupies these areas, the light blue occupies these areas, and the granular occupies these areas. And you can see, and these are just slices of the brain, and this isn't by any means all the connectivity in the brain, but it shows that they go back and forth, sending prediction signals and prediction error signals. So this is the instantiation of this hierarchical predictive coding in this organization in the brain. And what this does is this is actually slightly different from what we talked about before. Because if we look into the mechanism here, we see that whereas before we had priors being suppressed from the top, and I'll just quickly run through this because you've already seen this before. And then when they're suppressed from the top, we get bigger prediction errors and large change. What's actually happening is starting from the bottom. And so the prediction error neurons are on the right-hand side. And these are the neurons predominantly located in the correct laminar layer that have the 5-H2A receptors. And when these receptors are bound by LSD, as we just learned from Asher, they become more excitable. They fire more. And so we're starting with receptors that are in the prediction error neurons, and they're firing more. So we're starting at the bottom. And so they're sending bigger prediction errors. Even though they're being suppressed from the prediction neurons, they're more sensitive just in general. And so any minimal information coming through is making them more excited. They're sending up increasingly large prediction errors. And then this causes a bigger change, even in the very strong bias. You're getting so much prediction error coming up that this starts suppressing the posteriors, and then we get bigger and bigger change. And you can see what happens here. As these priors go down and down and down, you get a flat energy landscape. Your senses open up and you send even bigger errors, less and less priors, bigger errors, less and less priors. And you can see how this is kind of a feed forward loop. You get more and more sensitive to sensory information as your prediction errors get larger and larger. Right? So this is really, really neat. To summarize, what's happening here is increased sensitivity to sensory inputs. You could call that openness, makes you less confident in your prediction bias. Right? Another way of saying that is in the face of sensory overload, which is a common terminology for psychedelics, we can call that new information. You question your strict model of reality, right? So just the simple fact that you're now more sensitive to real information in the environment makes you less confident in your model, as you less biased. This is pretty profound in and of itself. We have this hierarchical predictive coding. We have this new mechanism of how sensory information propagates up to reduce our certainty, but we still haven't actually shown that there's evidence for this. We've just now instantiated this theoretical model in a biological model. So let's look at this in more detail. Here again, we're looking at the laminar layers and the cortical slices, and let's do some tracing of neuron activity to see what happens. This black line here is showing sensory information coming from the thalamus. The thalamus is kind of like the gatekeeper of sensory information in your brain. It screens most sensory information except olfactory information, and it's going to send up this information to these prediction neurons. This isn't strictly what's happening in the brain. It's obviously very, very complex, but for the purpose of this presentation, we're going to simplify it. Then these prediction neurons send predictions, and then these prediction error neurons send prediction errors back, right? It's this loop we had before, and they're sending back and forth. And so in these brain areas, what we're seeing is this communication back and forth, back and forth, back and forth, back and forth. And you can imagine that across an entire brain, these back and forth, back and forth, back and forth signals might actually start looking like large-scale activity that we can measure. And indeed, that's what we see. I'm not sure how this is working out on Zoom, but what you should see here is a brain with slowly changing brain activity, right? This is actually a functional resting state, fMRI. And so what we're measuring is a proxy of brain activity here in someone who's in a default just resting state. And in the paper, they talk a lot about the default mode network. We're not going to get into too much detail here, except suffice to say that in your kind of baseline activity, your brain goes through this kind of back and forth prediction activity. And what characterizes this back and forth activity is an activity pattern called the alpha band. So these are different types of brain bands, and they all overlap. It's all a spectrum, but we can isolate this certain frequency called the alpha band, and this is thought to be this baseline activity. And what the REBUS model says is that high alpha activity would correspond to a state of low entropy, right? That is to say that these alpha activity bands here correspond to being in the trough of an energy landscape, so a stable landscape where you have more order and you have strong priors. And so then what would we predict is that under psychedelics, when we take a psychedelic, the energy landscape will dissolve, it'll become flat, and then we'll get desynchronization of the alpha band. So what this means is that we should be able to measure a decrease in alpha activity with certainty, but it would support the hypothesis. If we don't see this, we need to refine our model. But we do see this activity. What you're looking at here is the average of brain activity of psychedelics versus non-psychedelics. And anything in blue shows a decrease in activity. And these rows here are the different brain frequencies. These are two gamma bands, beta, alpha, theta, and delta. Okay, we're not going to worry about too much about the other ones, but what we do notice here is that, remember, blue means decreased activity compared to normal, right? We see that psychedelics LSD, psilocybin, and ketamine, they're blue, and LSD is bluer than most. And this might have to do with the fact that it's that much more potent, like Astrid just talked about. And you'll see that actually LSD decreases across all bands, not even just alpha, whereas psilocybin is mostly focused to alpha and beta, and ketamine is mostly focused to alpha and beta. But at least with the REBUS model, alpha and beta are significantly decreased with psychedelics. So this confirms that psychedelics do have this effect. This is very interesting. This is supporting the model. It's like binocular rivalry. What happens is if you picture yourself looking through a pair of binoculars, what happens is in one eye, they present one image, green slanting to the right, and then the other eye, we have red slanting to the left. And what your brain will try to do is it'll try to just see one image. And so if you think about this being over time, you start off seeing just red, then you go green, red, green, and your brain switches back and forth. And it does the switching very quickly. And the interpretation of this is that you have these very strong top-down priors saying there is only one reality. And so it tries to pick one or the other, and it can't decide. So it switches back and forth very quickly. If you take something like psilocybin, which they did in this experiment, then what happens is the switching becomes much more slow. It becomes much more stable. Your brain is more comfortable being one or the other. It doesn't feel like it has to go back and forth as quickly. And furthermore, unusually, it's comfortable seeing both at once. Normally, it's not comfortable seeing both at once. It wants to pick one or the other. But here you can see what are called mixed precepts. You see just a total overlap or kind of a mix of the two. And the interpretation of this result is that because you have these weaker top-down priors, your brain doesn't say, well, it has to be one reality. It's like, well, let's just see if it's whatever it is. You have more sensory input, let's just see what it is, instead of imposing a top-down prior on it. This is very interesting, and this might explain why you have hallucinations. You're getting sensory input from, like, say, audio, well, see what this looks like, and it actually puts it into your vision as opposed to just eliminating it before. And so this could be a possible mechanism for that. So let's go over quickly what we've talked about so far, because we've already talked about a lot. We now know that with our hierarchical predictive coding, that in your normal resting state, you have algorithms that define this default mode network, okay? This is a state of more order of strong priors, right? When you take a psychedelic, you have less order and you have weaker priors, and this leads to a desynchronization, which you can visualize, and you can also measure with like a behavioral task like binocular rivalry. And say, okay, that sounds all very interesting and very cool. When you're under psychedelics, you have these proposed effects and we can measure them, that's neat. But aren't there these long lasting effects that people experience? How can you go from this like transcendental experience to just, why don't you just go back to your regular beer drinking self afterwards? How do you retain a benefit? And one answer to this is that psychedelics increase plasticity. It's not the only answer, but one of the answers is that they increase plasticity. So if you remember the neurons from before, this is a cartoon neuron, right? You have your cell body and you have these dendrites, these neurites. And on the dendrites, if we zoom in, we have these dendritic spines. This is a normal neuron, looks pretty healthy. It's got a couple of them. But under psychedelics, what happens is you get way more neurites, so you get more projections coming off it, and you get way more dendrites. And this isn't just a cartoon. So if we look at real microscopy data, we see this is a regular neuron right here. You know, this is a neurite, doesn't have many dendrites on it. And then we have 3 different psychedelics, and you can see that there's actually many more projections. Anything in orange is stating actin, and actin showing projections coming off. And we can see, especially LSD shows many, many more dendrites coming off. And in this paper, they include cartoon diagrams of what these neurons look like. And these aren't just random cartoons. These are actually representative tracings of cortical neurons. So these are kind of average tracings of neurons done with microscopy. And you can see that here's a healthy looking regular neuron, but with psychedelics, they become much, much more branched and become quite complex. And all this extra complexity is extra connectivity. This is real connectivity happening inside the brain. So your brain is rewiring itself under psychedelics. It's not just a transient experience. And you can measure this neurogenic efficiency. And they do this in this paper with this number of psychedelics. And what you're looking at here is this is a concentration of the drug. This is the efficiency of neurogenesis, okay? And they use ketamine as the reference point. Ketamine is set to 100%. So anything below 100% is weaker than ketamine. Anything higher is stronger than ketamine. And you can see that all these drugs have very high neurogenesis above baseline, right? They're all higher than the normal. But MDMA in particular is extremely high. It's more than twice ketamine and way higher than everything else. And one possible explanation for this is that MDMA is not just a 5-HT2A receptor agonist. It's just a general serotonergic releasing agent. It's also dopaminergic. It's kind of the whole gamut, right? So this might explain why it's much more potent. Just an interesting side note. And in this paper, they put forward a hypothesis that I'd never heard before, and I think is pretty fascinating. And it's not necessarily their work, but they're pulling into their work. And that's that they think that the serotonin system is an adaptation system. And so what happens is we walk around these very, very biased, strong models, and they're very resistant to change. But under certain circumstances, you need to change this model very quickly and very dramatically. If you have this belief that lions are very friendly and you should go and pet them all, and then you go up and pet one and it almost kills you, you need to revise that belief immediately. That can't be something that you keep trying over and over again just to make sure about it, right? You need to make that change automatically. And so there needs to be a mechanism to fight against this really, really strict repressive bias that your brain has. And they think that serotonin is this system. And the interesting thing is that, okay, that makes sense under a normal circumstance, but under a circumstance where there is no threat, where there's nothing to actually adapt to, then what happens? And so the way I think about this is that instead of the model adapting to the external world, it has nothing else to do but adapt to itself, which is an interesting thing to do. So that brings up the question, how does it know how to adapt? What is it going to adapt to? If this is our energy landscape and we're in this current state and we want to get here, how do we do that? How does it know the right way to do that? And the answer, the authors think, is insight. So this looks like a complicated graph, but we're going to break it down and we're going to only focus on certain points. The black line here is a controlled drug called DXM, okay? It's not a psychedelic, it's kind of related to opiates, it's in cough syrup. It's a good control because it makes you feel weird. As you can see, it makes you feel disembodied as the disembodied. Anything closer to the outside is a stronger effect. So it makes you feel disembodied and some other things. But you can see that psychedelics really have much more insightfulness. This is compared to baseline as well. And so this isn't just unique to this experiment. Psychedelics are known for their insight. People talk about these life-changing experiences. This is a much known anecdotal thing, but we can also measure this experimentally. And the mechanisms of insight are actually quite well known from the literature on learning. The first step is you need a curious behavior, some kind of exploratory, novelty-seeking learning. You have to want to learn something. And this is the analogy in a therapeutic setting to set. You need to have the right set, the mindset. You need to go in wanting to change. And the second part is more complex. It's called structure, fact-free learning, abductive reasoning. And for our purposes today, Bayesian model selection and Bayesian model reduction. And this is a really, really fascinating concept that blew my mind when I learned about it. And we can think about this more as the setting. You have to have the right environment to create a new model. Okay, so the first one's relatively self-explanatory. We understand you have to want to learn something, but let's dive into more this Bayesian model selection and Bayesian model reduction. So in the paper, they define Bayesian model reduction as the mechanism which high-level models are stripped of their redundancy so that simpler, more refined solutions may be revealed. What does this mean? Okay, so this is actually taken from a paper on Bayesian model reduction. This isn't just some random image. And this is a model, okay? We have a bunch of nodes. These circles are nodes, and all of them are connected to everything else, right? It's a complex model. Everything is connected to everything else. It's actually hard to know what direction anything moves in. And instead of just being the numbers, let's populate this model with things we understand. Let's just say 1 is stress, 2 is increased heart rate and sweating. We think it's associated with stress. And then anxiety is kind of lumped in there too. And then on the other side, we have withdrawal, loneliness, sadness, and depression. So we have all of these, physiological symptoms, feelings, experiences. And in our model, we might think, oh, well, all these things always happen at once, and I don't know how they all work, and everything just connects to everything, and everything causes everything else. It's just total chaos. My life is super stressful, right? We call this model is too complex. And in a mathematical sense, we call this model, it's overfitted. I'm not going to go into what overfitting is here. It's a very neat mathematical concept, and it's simple to understand, but there's just not time for it. But this model is, it's too specific in its prescriptions, and it doesn't generalize well, and you can't actually figure out what's going on. Okay, this is analogous to being stuck in one certain state. Being in a deep energy landscape isn't necessarily wrong. It's our default state. But if you're stuck in the wrong part of this deep energy landscape, then it's a problem. And let's say you are, if you're in this too complicated model, right? So then you go and you take some LSD and you flatten your energy landscape, you weaken your priors. Then the connections between all these nodes become much, much weaker. They're no longer as strong anymore. You have these weak priors, and these nodes are just floating around, and they need to be reconnected again, okay? In the paper, they call us an entropic hot state. Everything's connected to everything else. There's lots of communications. All your presumptions, all your biases, they're much, much weaker. You can actually measure this entropy in the brain. And so you can see that the blue line is placebo here, and psilocybin is the red line. And it's higher. This is the entropy of the brain. It's a lot more connected. It's a lot more hot. There's a lot more going on. So you're in this entropic hot state, the flat energy landscape, and you start moving around. Instead of just being stuck in your hole, you're going to explore different places. And what you're doing is you're actually exploring different models. As you move around, you're exploring different models. And there's a quote that's attributed to Albert Einstein. It goes something like, everything should be made as simple as possible and as complex as necessary. So when you're building a model, it needs to be as simple as it can be, but it also needs to be as complex to capture everything you need to capture, okay? So let's see what might happen if you're exploring this energy landscape. You build your first model and you say, well, I'm gonna connect these 4 things. That's simple, but look, we're missing any connection this side, so that can't be right. Let's add some connections, and you can see that you're going to go through building this model, connecting different things, trying out different things. Now, it's not like you're doing this explicitly. You're not like, oh, I have this model, I'm going to build it in this different way, like you're building Lego. But your brain is subconsciously going through and trying to refine these models on the psychedelic experience. And as it keeps trying new things, it discovers, wait a minute, I have this model. Everything's connected, and it's much simpler. Maybe this is what's happening. And then this model solidifies as you leave a psychedelic experience, and then you end up in your energy landscape, but now you're in a different part of the energy landscape, so you have a simpler model now. And there's a much more prescriptive understanding of what's happening. Oh, look, I have a stress, and this side is stress, and this side is like a depression and emotions. And what connects them is the anxiety. My anxiety causes my withdrawal because of stress. It's not everything's connected to everything, it's just total chaos. It's very simple understanding of what's going on. And now with this understanding, we can actually change our lives, right? We have a new simplified state that's much more accurate in the long run. And what this type of learning is called fact-free learning, which when I first heard it was like, well, that's the most anti-scientific thing I've ever heard. How could something be fact-free and be effective? But that's not actually what it means. What it means is that you don't need new information. You're working within yourself with the information you already have, and you're simplifying your model with the information that you have. And so you're like, okay, well, that sounds cool, but does it actually work? And the answer is it remarkably does. What we're seeing here is we're seeing improved accuracy with psychedelics. These are people who have treatment-resistant depression. This is depression that doesn't respond to any other drug or any other treatment, right? Nothing works for it. And these people show increased accuracy with their predictions after psychedelics. This is pretty incredible finding, right? They have these models, they're super stuck. When you're in this depressive state, we can hypothesize you have really, really strong priors, everything's against you, you're really, really stuck, and somehow we can improve their accuracy of life events after psychedelics. That's remarkable. And likewise, we can also see that we reduce cognitive bias. So this is just another measure of model accuracy. So here we have a reduction, and 0 is complete accuracy, right? And you can see that they're very, very close to that. You get much more accurate. And so we have evidence here that this Bayesian model reduction is happening. It's a mathematical phenomenon, and it's a model just like the hierarchical predictive coding, but we have biological evidence that this is true. It's fascinating. A little bit, this topic of criticality. And it's just so neat that I want to cover it here. So when we're building new models, we're in a psychedelic experience. There's lots of information, lots of connectivity. And wouldn't it be great if we could process that information faster? And what is interesting is that if we're in a critical state, the answer is we can process that information faster. So not only do we have more, we can process it faster. Criticality is actually a concept from thermodynamics. And the analogy that's often used, because it's a literal example as well as an analogy, is heating up iron. So in iron, you may or may not know, there's iron atoms that have a certain polarity. And when they all point the same direction, such as this one, the iron is magnetized, all right? And if you take a heated piece of iron and you cool it down, what you might see is that slowly, I don't know if this animation is showing properly, over time, these blobs become more and more and more condensed as the neighboring atoms turn the other ones in the same direction as them. So they have a magnetic polarity, and they kind of influence the neighbors next to them to turn to the same one as them. And so over time, this whole thing becomes the same color. Whereas when it's really, really hot, there's so much energy that all the atoms are just spinning in all different directions, and there's no correlation at all. It's just total random noise. But in the middle, at the critical temperature, what happens is they start almost communicating with each other. And even though it looks static, like here, you can see that there's these blobs that kind of form and unform, form and unform, form and unform. And what's happening here is they're actually incredibly sensitive to very, very small perturbations that cause them to interact with each other and generate these complex patterns. And remarkably, people think that this is how the brain works as well. So let's actually look at these images. It was reported before that when I play all these at once, it doesn't actually play very well. So I'll start playing the brain first. And if you can see the brain, you can see these kind of slowly undulating patterns. They're not static patterns, they're slowly changing. And if we look at the subcritical regime, we say, well, it kind of looks like it. There's these blobs. But you'll see these blobs just slowly disappear and become more and more concentrated. They don't actually evolve. Whereas the blobs here are evolving, and they're moving around, they're changing just like they are in the brain. Whereas here it's just total chaos, and we don't really see much happening at all. It's just madness. And so the question is, does the brain operate like this? Does it have this critical feature? And one way to measure this mathematically is with the power law. I'm not going to go into this at all, except suffice to say that the interactions between these different things can be measured as a power law. And so if you measure brain activity and it matches a power law, this is evidence saying that it's operating near criticality. And this is actually what we see. So a normal brain operates near criticality. So these lines here are power laws. And the brain activity is in light blue and dark blue. So dark blue is regular brain activity, and you can see that it's pretty close to its power law. And light blue is psychedelics, the LSD here, and it's also pretty close to its power law. And you say, well, they look almost exactly the same. How's there any difference? But if you take a refined measurement and look what are called residuals, you'll see that, and what you want to see in your residual, this is the residual plot here, is you want to see them as close as possible. You want to see them as small number as possible. And so you'll see that LSD in every case is much smaller than the placebo, meaning it's much closer to criticality. And the really interesting thing about criticality is that at a critical level, you have maximal information processing here. So if we look at this plot here, this is criticality. And what we're looking at here is a model between excitation and inhibition in neurons. And you'll see that if we have the wrong excitation and wrong inhibition, if we're too low, we don't have a critical line. And if we're too high, we don't have a critical line. But if we have just the right amount of excitation and inhibition in the brain, we have this critical line. And furthermore, when we measure the amount of information processing possible at these different levels, we see that it peaks at the point where we're at criticality. And what this means is that the more close you are to criticality, the more optimal your information processing is. And this is what they think is happening in this entropic hot state. Everything is connected, and not only is it all connected, but you're processing information at a more efficient way than you ever could before. Pretty incredible. So let's summarize this. We have our model. This is our new refined model. This model is actually real. It has an improved accuracy, and this improved accuracy might be mediated by the fact that when in a critical state, we're processing information more optimally than we would before. Pretty fascinating. So let's put it all together. Everything that I've tried to talk about in the REBUS model, we're going to try to throw it together in a couple slides, okay? Right now, this is your current state. This is the energy landscape of your brain that you're in. And you're here, and you want to get to a better state over here. Right now, you're in an energy minima, not the best one. You have your alpha activity as your default activity. You're very structured and ordered with a strong prior that suppresses you from adapting to new information. You're biased. And you have an overcomplicated model, okay? How do you get over here? Step one, you have these strong priors that are suppressing every layer down, right? I just want to walk through this hierarchical model again because we're going to come back to this. This is kind of the crux of it. And these strong priors are suppressing their prediction errors, which makes you biased. And you have a very small change in your opinion after new information. So then you take LSD. LSD binds to the receptor in your brain, 5-HT2A. This causes the excitation of pyramidal and neurogenesis at the same time. This increased activity in prediction error neurons are on exactly the right type of neurons that when they increase their activity and are in the right type of the brain in the right areas for the hierarchical predictive coding and abstract reasoning, they cause increased air propagation up from your senses. So even though they're suppressed by high-level priors, they're still so sensitive, they're activated by psychedelics, they send up increasing prediction errors from your sensorium. So despite the fact that there's strong priors, you have weakening priors at the very, very top, a large change, and then this causes weakening priors that percolate all the way down the network. And as these percolate down, they then reach the senses. Your senses are less suppressed than before, which causes you to have even bigger prediction errors coming up this side, and the cycle repeats itself over and over and over again. You're becoming more and more sensitive to information. So you start off with more order, deep energy landscape, alpha bands, and a complex pattern, and then you go to less order, flattened energy landscape, desynchronization, and you lose the connectivity between your model. And then this is where the magic happens. With a deconnected model, you have a critical network that's processing information optimally. You start moving around in this energy landscape while you're processing information optimally, and you find out a more optimal model that allows you to be more peaceful and more understanding of your environment. And with the neurogenic actions, this effect lasts. It's not just transient. And then you end up in your new state. And remarkably, this new state looks very much like the old state. You have you're in your minima, you have alpha activity, you have more order, you have strong priors once again that block new information, so you're biased. But instead of having this overcomplicated model, you now have your new better model. And that's what you're left with. You're left with a new better model, a better understanding of reality. And this is the takeaway from the paper, if there was one for me. They say, thus, a commitment to the discovery of better models is important, as improved models will provide more robust and reliable approximations to reality, which are likely to support greater stability in terms of mental health. And this is a cartoon. It's a favorite cartoon, and it ties so well to the situation and ties well just to life in general. When we commit ourselves to new information and making better and better models, we just have a better experience and we're better people in the world. It's just an all around better experience because you're more accurate. You'd have a better idea of what's going on. And with that, I'd like to acknowledge science because it's just amazing. I mean, in this presentation, I was searching things and all the answers were there. It's just incredible. These are all people doing research, cranking it out. It's just amazing what we know now. And Google for helping me find it all. I mean, it's all there. Google image search is amazing. Carhart, Harris, and Friston. I mean, this is just an incredible piece of work. It's been amazing for me to read. I like to thank my wife, Emily, because I have literally dedicated so many hours to this, and so I thank her for her tolerance. I'd like to thank friends and family in the community for tuning in. And finally, MAPS Canada Journal Club for giving me this opportunity. Thanks so much.
All right, thank you. We have a couple of questions, and there's not that many, but they're pretty complicated. Can you first off, you cut off a bit during the mixed precepts with sensory input leading to psychedelic effects. Would you be okay going over that again?
Oh, the binocular rivalry.
I think it's
here?
one more, yeah, that slide that you're hovering over. Yeah, that one.
This one? Do you see this now? You have the full screen?
Yeah, it's just your audio that lagged a bit.
Okay. Yeah, so what's happening here is there's an experiment where each eye is isolated from each other. It's like looking through a pair of binoculars, except at each end of the binoculars, a different image. And so in one eye, you're seeing one image, and another eye, you're seeing another image. And what happens is because of the strong bias of your top-down priors, your brain really wants there to be one reality. And so it switches quickly between one image and the other image. Like your perception, your image that you see is one or the other. It's interesting. You don't see a mix of both. Your brain forces one or the other. But under a psychedelic like psilocybin, what happens is you have much more stable switching. So instead of it switching quickly, your brain can't decide. It wants one. Your brain's like, okay, well, we'll accept this for a while. And then, okay, we'll accept this one now. And also what's interesting is that it allows mixed precepts. And so this is where you actually see both images at once in different forms, either fully overlapped or just partially overlapped into kind of a mix. This is something you don't see that much when you're not under psychedelics. And the interpretation is that your brain, because you have less strong priors, you have less of a bias in what you want to see, your brain allows more of a mixed experience to occur. And then I also tie this into the possibility, this is why you might hallucinate under psychedelics, because it's different sensory information is now going to your visual cortex, which normally would be suppressed because it's nonsensical to your visual cortex, but because you're more open to new experience, you actually see these different sensoriums like sound or tactile information.
Thanks. And following from that last question, does that concept explain synesthesia or some other effects of autism?
Yeah, so I think the short answer is possibly. There actually is a section in this paper on other psychological conditions, and they do mention autism. And I talked about that actually in my last talk a little bit. It's a little bit of a longer answer, but I suffice to say that yes, there is a connection there, and I encourage you to look into it.
Another question was, how long does a state of increased plasticity last under psychedelics, and do the changes remain indefinitely?
Oh, that's an interesting question. The short answer is I don't know. The plastic state doesn't last much longer than the serotonergic activity of the psychedelic itself. There is some the genetic stuff that happens a bit longer because it has to cascade through the cell, but it's not. But it looks like it's probably on the order of hours and possibly days, depending on the psychedelic. But obviously, it's decreasing as the concentration goes down and your brain adapts. And is the change in your model permanent? Well, nothing is ever permanent. I mean, you're never stuck. Your model is always changing and updating, even in incremental ways. So nothing is ever permanent. It's something that you've learned. Insofar as learning is permanent, it's permanent because it's just a learning. But it's not like it can't be changed again.
Another person asked, or they said that some papers that they've read have mentioned that meditation has a similar effect on the default mode network and alpha wave activity. Is this relation touched upon in the REBUS paper?
It is actually. So in the sections of different psychedelic states, that's one thing they talk about. So they have autism, they have schizophrenia, they have depression, I think they have anxiety, and they might have eating disorders, and they also have meditation as well. And so yeah, there are some overlaps in that. Obviously, it's not to the same extent, except if you're very, very experienced. But yes, there is overlap.
All right. And last question: how do bad trips relate to the criticality network? The criticality theory applied to the brain. Would variants in genetics, such as types and quantities of serotonin receptors, explain such experiences with psychedelics?
So I don't think criticality is especially pertinent to bad trips. Bad trips are more have to do with the set and setting that you're in. And so yeah, you're in an enhanced processing state under criticality, but that doesn't necessarily mean that you're going to go bad trip or good trip. It just means that in the set and setting that you're in, you're going to process it optimally. So I don't think there's necessarily a connection to bad trips there. In terms of psychedelics and your genetic predispositions, I mean, there are known variants in receptors that have to do with depression and anxiety. I mean, there's like the TRAC2 receptors, the BDNF, which is a neurotropic factor. There's serotonergic variants as well. These have to do with susceptibility for sure. And so I don't actually know how these same receptors though correlate with psychedelic experience or experiences afterward. That's gonna have to wait for further research.
All right, thank you. Those are all our questions. And thank you so much for the amazing talk.
Thank you so much again for giving us this chance.